Alright. Let's see what happens. I'll get that off. And we will go here. Okay, let's see what happens. The picture quality is actually better on Facebook Live. Maybe YouTube needs to go and sit in the corner and give itself a talking to. Ugh. Oh, cold coffee. Ugh. That's terrible. All right, so where was I up to? 11 people back in already. Guys, thank you so much for bearing with me. Thanks for giving me the feedback. We will ditch YouTube Live from now on and just do Facebook Live, and we'll continue that way until I find a better option, essentially. I'm at 30 frames a second at the moment, so let's see what we've done. Oh, Jesus, we've got 20 people watching. 22. Wow, okay, right. Definitely YouTube was a problem then. Okay. All right, I'm hearing you guys loud and clear. That's fine. That's okay. All right, so right, so metabolism of nitrifying bacteria. So that's essentially what they do. They take ammonia, they take nitrite, they oxidize it into nitrite or nitrate. You get varying differences on that. That's essentially what they do. They have a very specific function. They don't really deviate from that path. The types of nitrifying organisms that we have, however, is very varied. Now, nitrifying organisms in particular, the old Nitrosomonas and Nitrobacter strains, they were usually isolated from wastewater. And those guys really like high levels of ammonia and nitrite in the water. They thrive on really high levels, and I'm talking over 20 parts per million of ammonia and nitrite. And that's where you'll see those guys really thrive. In wastewater applications, in water that's got just really shit-ass water quality, essentially. So you don't usually find those in mature aquaculture or aquarium systems or life support systems in general. Now, you would remember, I'm going to say maybe 5 to 10 years ago, Dr. Tim Hovenick isolated Nitrospira and Nitrosospira bacteria as what he believed and he found to be the dominant species of bacteria in aquarium biofilters at lower levels. So lower residual levels of substrate. And by substrate, I'm not talking about gravel. By substrate concentration is what I'm talking about when I talk about ammonia and nitrite. And that's what they call a substrate concentration. Okay. So if I use that in the future, you know what I'm talking about here. So when he found those and they started growing them, that sort of produced flipped this whole thing on its head, right? Now, I believe David, I can't remember his last name, he was putting up some information on the Q&A about some archaea. And strangely enough, archaea is something that I know quite a bit about, probably not as much as him. He's done quite a bit of research on it. We talked over a couple of emails, actually. And archaea are really the primary kingdom of microorganisms before you sort of even get to nitrifying organisms. And these guys can directly consume ammonia and nitrite and all other manner of waste products. They can directly consume it, or they can act as sort of something like a living enzyme existing within the biofilms with other bacteria in general, breaking down the product and feeding it in a much more, I suppose, broken down, palatable, more easily consumable form to accelerate waste reduction, essentially. Now, archaea was something that we were looking into and researching, and we currently still are. The archaea that we were using was being marketed directly towards wastewater plants in quite high concentrations. Now, the power of archaea is not to be underestimated. Archaea generally is considered as being in very low concentrations within the environment. However, I believe it was David, and my apologies if I've got your name wrong. I can't look back on the conversation at the moment. But essentially, the diagnostic measures that were being used weren't actually targeting or able to actually identify archaea within the aquatic environment, which is why maybe there's been an argument towards them not being in high concentrations within the aquatic environment in the past. So when we're talking about archaea, one very impressive example that I was given by the manufacturer was a wastewater treatment plant in his local area overseas in Europe was having problems with residual ammonia levels above 30 parts per million, and they were really getting hammered by the EPA as to their discharge levels. And they couldn't change anything within their system. They couldn't change their biological filtration processes. It was an aging plant, an aging wastewater treatment plant. So they started adding this archaea product to the water. Now, as I said, archaea acting as a living enzyme, and it dropped the ammonia levels from over 30 to 50 parts per million to below 0.25 parts per million within 5 days, and this is in a 120 megaliter a day wastewater treatment plant in a small village. Like 120 megaliters for a wastewater treatment plant isn't really that big. It sounds like a lot. 120 million litres sounds like a shitload of water. But when you're talking about a wastewater treatment plant, it's not. When people poop, there's a lot of it. So, it's not that big. But having said that, within 5 days, dropping it from over, when they couldn't get it below that for the last 20 years, and it's dropped it from over 30 to 50 parts per million to 0.25, and then kept it there for over 6 months, when they've got constant flow through of effluent going through there, was really impressive. They're very persistent organisms, and if you can grow them in high enough levels, they can actually be very, very beneficial. So that just goes to show that our technology and our understanding of these nitrifying organisms is constantly evolving. And if you don't educate yourself, you're going to fall behind. So nitrifying organisms can also be heterotrophic as well, which I'll get into. They don't do it as effectively as autotrophic nitrifying organisms do, our normal nitrifying bacteria, but they can do it. Generally speaking, the water quality ranges that these guys actually live in, you want to, ideally the pH really needs to be above 7. They don't like an acidic environment. You need alkalinity within the water, so your water needs to be buffered. You need to have a minimal alkalinity within your water. If your alkalinity drops, not only does the food source that I was talking about with the nitrifying bacteria, the alkalinity that they use, not only does it starve them out because they can't access that food source, that nutrient anymore, but the pH drops, and when the pH drops, they don't like that. When they've got no food, they essentially die, and your biofilter dies off. Now, I can probably bet that anyone with a store that's tested anyone's water, and they've come in and they've tested pH, and just like the last time I went through it with you with water chemistry, you should also be testing KH as well, or alkalinity. I can almost bet that every time they had a low pH problem, and a ammonia spike, there was no carbonate hardness in the water or no alkalinity in the water. And the two prongs is that your pH drops, your bacteria don't like it, and your alkalinity drops, and therefore they have no way of actually processing the ammonia and nitrite, and therefore they die off. Zoe, what's classified as low alkalinity for them? 0. As long as there is some alkalinity in the water and the amount of alkalinity within the water is able to sustain itself over a period of time so that it doesn't drop below 0, you will continue to have metabolic action within your nitrifying bacteria. I'm talking predominantly freshwater systems. Saltwater systems are never going to have this problem because they've got calcium carbonate substrate. They have higher alkalinity levels in general. So, but in a freshwater system or an aquaculture system or something like that, but generally speaking, in an aquaculture system or an aquarium system, you want to maintain alkalinity over about 20 to 30 parts per million as a minimum. I used to aim for 50, and if I needed to buffer it down from pH for low pH species like tetras, I'd use pH down. It's not ideal, it's got phosphate in it, but it works. For higher pH species like guppies, live bearers, cichlids, it's never a problem because you're having it over 100 parts per million, and even goldfish they like their pH really high. So high pH actually is a way of knocking out pathogenic bacteria as well. So yeah, you're never going to have a problem in a saltwater system. Alkaline pH is a problem. Low pH is a problem. Goal range for alkalinity above 0, and the range that you would have would be totally dependent on the species and their requirements for pH. As I said, you can buffer that alkalinity up and then bring it back down with pH down while still maintaining that alkalinity level. But the pH down, the sodium biphosphate will buffer that pH down without actually removing the alkalinity from the water. Okay, so temperature is a big one. Above 25 degrees, you're going to have really good metabolic action within your nitrifying bacteria. Below 20 degrees, you get about a 30% drop. Below 15 degrees, it's almost a 50% drop. And once you get to 8 degrees, you're really having to pretty much give them a walking stick to get around. When they get cold, they're just like us. They don't like being cold, and their metabolic process drops. So temperature, the higher the temperature, the faster you're going to oxidize ammonia and nitrite, essentially. Nitrifying bacteria don't take a lot to look after, and it constantly surprises me how often people screw it up. Which is why not only we've developed these bacterial products, but also we're developing a protocol now for cycling aquaculture systems and biofilter systems, which is where we're getting all that really useful data from. Isolating and growing nitrifying bacteria for commercial use. So we went through a heap of different biofilters, different substrates from aquariums, aquaculture systems, all sorts of stuff. And we found some really interesting stuff, like I said, strains that oxidize not only ammonia but also nitrite simultaneously. Stuff that oxidize nitrite with no accumulation of nitrate. Some of these strains are denitrifying nitrate, which we were just really surprised at. These strains are they're not now only isolated to autotrophic bacteria. Some of these strains that we isolated, we believe are heterotrophs, or they're opportunistic heterotrophs, so they can do it autotrophically as well as heterotrophically. The way we grow these and then the way we nurture them are two completely different systems that we use. We use, different nutrient sources to grow these than what we do to actually nurture them within an aquatic system. So it's mind-blowing what the things that we found. Archaea, for one, it's getting out of the mindset of saying it has to be a Nitrosmonas, Nitrobacter, Nitrospira, Nitrosospira, Nitrococcus, Nitrosococcus type of autotrophic bacteria is just throw it out the window now. We don't adhere to those rules anymore because of all this new information that we're finding. So, and this is constantly evolving. As I'm saying, we're constantly looking at new strains. We're constantly doing research and bench testing on these guys. We're currently doing DNA tests and PCA tests on the different enzymatic processes to actually nail down further exactly what these strains are. When you do a DNA test, you get a match. You might get a 99% match, but in that 99% match, there still might be 100 different strains of bacteria. You can have a pretty good guess what they might be, but when you're mapping the genome of a bacteria and that specific sort of bacteria has, 100 related species that all share 99.998% of the same genes, but that 0.002% of genes can mean the difference between it being useful or completely useless. It's really hard to nail down a strain for bacteria. I've got to tell you, it's big work. Does the bacterial action follow a linear metabolic rate curve? No. That's got to do with the substrate concentration, alkalinity, temperature. You can see it follow a trend, but it's not a linear curve. In no way is a linear curve, and that's why we're trying to optimize the growing and the maturing of biofilters with these new things, with putting together a program to supply the things they need on a consistent basis. You miss 1 or 2 days in ammonia dosing, like ammonium chloride dosing for fishless cycling, and it can set you back, it can set you back a week and a half, maybe more. You've got this bacteria climbing, and then all of a sudden they go, oh, well, I've still got 4 parts per million of ammonia in my system, so I'm just not going to dose, when they've been adding 100, 200, 300, 400 grams of ammonium chloride. It's not like that. It's more like 20 gram increments or something like that, depending on how much bacteria you've added to begin with and how fast it started up. But you're climbing, and then they say, oh, no, we're not going to do that. So the bacteria are just going to consume the residual amount of ammonia in the system, and then 3 days later they come back and they go, oh, well, it's at 0. But within the first 24 hours, they've consumed all that ammonia. So they've had no ammonia for 2 days. And then all of a sudden you throw ammonia in the system and throw a residual ammonia level of 4 to 5 parts per million or maybe more at them, because consumption is now outstripping concentration. It's like getting kicked in the sack. You got to look after these guys. The thing is that in a stable, continuous culture system, you've got a continual amount of ammonia production and a continual amount of food going in. When you're trying to nurture these guys to grow them in a biofilter, you're tipping in, a CFU concentration of maybe 100,000 CFU per mil to actually culture the biofilter. And then you're throwing nutrients and trace elements and stuff at them to get them to grow. And all of a sudden they're just like, whoa, I've just been thrown into this entirely new environment. Now I've got to find a place to live. I've got all this aeration and water movement happen around me. I'm getting flogged through pumps. There's foam at the top of the water. I've got to find a place to live on this new plastic media, whereas I was just floating around happy as Larry in a bottle before. And now there's all this ammonia that's, crushing in on me that I've somehow got to find a way to eat whilst floating around in the water and trying to find a place to live. You got to be careful with these guys. This is why so many people have problems cycling a biofilter. We're back on. Yes, we are back on. Do the new strains exist in the aquarium if you do a fishless cycle with no bottled bacteria added? And didn't realise that the daily dosing was so important. I'm specifically talking about nitrifying bacteria, and I'm specifically talking about maturing a brand new filter. And in an aquarium system where you've got a residual amount of ammonia, the amount of ammonia, if it climbs to 4 parts per million and then drops to 0, is probably going to well and truly outstrip the production that you're going to have from the couple of fish that you ate at the start anyway. Okay, so don't get too concerned. I've got an entire calculator that will essentially prove that. In higher dosing applications, especially at lower temperatures, you do really need to be careful. The new strains that exist. Okay, new strains that exist. I assume you're referring to the strains that I'm talking about that we isolated. There is no guarantee that you are going to get those exact strains within your aquarium if you do it with fishless cycling with no bacteria added. No. The benefit of adding the bacteria in a bottle in the concentrated form is, one, these bacteria we've isolated and bench tested to do a really bloody good job really quickly. So you're not relying on native bacteria that might not be as efficient. Secondly, you're putting them in at orders of magnitude higher CFU concentrations than you would be waiting for them to develop on their own. Thirdly, the more bacteria that you put in there at the start, the more ammonia you can add, because the amount of ammonia in grams per cell, or femtomoles per cell, I should say, is going to be, collectively higher with more bacteria than it would be with bacteria that are going to, that are going to develop natively within your aquarium. Okay, so you're not always going to get the same amounts of bacteria. And are you going to have the same bacteria in there 2 months, 3 months, 4 months down the track after adding this bottled bacteria that we say is so good? Which, I mean, it's good. We bench tested it. We know it's good. We've got the data to prove it. Is that going to be there 2 to 3 months down the track if you don't redose it? Arguably, probably not. It's either going to mutate, or it's going to be, it's going to get replaced by another sort of bacteria as the ammonia levels stabilise and subside. The bacteria population within a closed aquatic system is continually evolving. And this is why continual dosing of probiotic bacteria, not nitrifying, because once your biofilter is established, as long as you look after it, you'll be fine. Unless you get a really old aquarium, it gets clogged up with crap and you need to reseed it just to refresh it, which is, not a bad idea in some circumstances. But this is why adding probiotic bacteria to it, or a natural source of water with natural diverse microbes in it, obviously you have to be careful of introducing disease that way, helps to maintain the biological diversity and keep things ticking over effectively and efficiently. Is it for marine only? No, it's for every single aquatic system. The daily dosing. Now I'm talking daily dosing. Again, that's for maturing a biofilter. Daily dosing for probiotics can be done. I would argue that it probably only needs to be done weekly to bi-weekly, maybe even monthly, and that totally depends on your system as well. Bruce, so given someone starting a fishless cycle on a reef and they have access to dosing pumps, would your suggestion be to dose your ammonium chloride via a metering pump? Yes, we actually did that recently at a prawn broodstock facility that we installed in Proserpine. We were dosing it for concentration to about 4 parts per million. And once it exceeded that concentration, we were dosing levels that would have resulted in 11, 12, 15, 20 parts per million, which obviously is not overly ideal. So we put it on our dosing pump that was dosing our, is now dosing our pH control for our alkalinity supplement, and just mixed it up at a concentrated solution and put it in and just adjusted the dose rate per day to put it in. And that way it doses it over a 24-hour period, which means the concentration doesn't exceed 4 parts per million or higher because it's being consumed as it's being added. But it also means that you're adding the required amount per day to effectively emulate the amount of fish that we wanted to put in there at stocking. These guys put in 750 individual broodstock prawns, like big bastards. And we needed to get that biofilter up and running and consuming as much ammonia and nitrite as possible before they did that. If they went and put those in, and it was a brand new biofilter, yeah, big dramas, big dramas. Bacteria psychology, yeah, either that or, grey hair. Oh, Tim, it would be a privilege. So that's nitrifying bacteria. I'm sure we'll come back to it when people have more questions. Probiotic bacteria. This is where we get into the meat and potatoes of what we're sort of talking about here. So we have a question. Is C-Chem Prime a good source to dose? Prime is an ammonia neutralizer. It is not an ammonia source. Prime is a chemical that binds up ammonia. Now, depending on what chemical they use, sometimes they're a formaldehyde-based product. Some of the more common ones are sodium formaldehyde bisulfite, so they do have formaldehyde in them. Arguably very low concentrations, so I wouldn't be too concerned about that. It is a very common chemical that you use in your ammonia nitrite, in your chlorine, chloramine, ammonia neutralizers. And apparently it's still available to the bacteria to eat. So if you have very high levels of ammonia and you've got fish in the system, yes, you can dose that in conjunction with water changes, in conjunction with trying to get that biofilter up and running by any means necessary. Just trying to reconfirm what I heard. Is there a maximum part per million of ammonia you should have when doing a bottled bacteria cycle? So I'm assuming if you've... Look, fishless cycle can be with or without bacteria. I would aim for a concentration to begin with of 4 parts per million. I would try and maintain 4 parts per million daily until you were testing 4 parts per million at the start of the day, and then at the end of the day or the start of the next day, it was very close to 0, and I'm talking 0 to 0.25. In an aquarium, arguably that would probably be well and truly enough if you were getting that sort of consumption to be able to add some fish to the system to, be safe. In an aquaculture system, it's going to go totally on consumption and how much ammonia we're calculating that we're producing and how much ammonium chloride we then need to add. It's very nuanced. But yeah, 4 parts per million, and make sure that you're 24 hours later. When you're at that point, you're good to go. Pretty much. But go slow. People use dead prawns to cycle tanks. Where's the problem? The prawn needs to break down. The prawn is filthy. The prawn is also better to eat than throw into a aquarium. I would probably argue that you would be better off, because you're going to have fish food available, you'd be better off to crush up some fish food between a couple of spoons and put that into the aquarium and let the ammonia rise. It's stuff that you're going to be adding to the aquarium ongoing. It has been heat treated. It's not going to have potential diseases on it. We all know what happened with the white spot disease that went through the farm that has since resurged again. Just don't. I mean, God, look, I wouldn't recommend doing it, but peeing in the tank would be safer. Urine is sterile. Urea will break down into ammonia. I have known people to do it, and it does do it very, very effectively. If in doubt, go and get some cloudy ammonia from the supermarket, or some ammonium chloride or something like that. God, if you're struggling for ammonium chloride, give me a call and I'll post you 100 grams, which will last you for, 10 years if you own aquariums. You don't need very much. There's not a lot of it in there. High dose of prime can restrict bacterial growth. I would definitely agree with that, Paul. And I would, in all cases, not use a chemical dechlorinator. I see too many problems with them. If they're overused, I would very much prefer to use filtered tap water that's filtered with chlorine. Filtered with chlorine. God, I'm tired. Filtered with carbon. Activated carbon. The prawn farm would have supplied its own ammonia source. Well, yeah, but the problem is, once the pond was cycled, they would have had no prawns to put in there. Can too much ammonia have an antiseptic antibiotic effect? Yes. Too much ammonia can kill the bacteria? Yes. Or it can fuel the prevalence and growth of other bacteria that you may or may not want in there. So, I think we've got over that. From what I can tell, Paul's probably still has his shirt on too, so we're in a good way. Metabolism. Okay, so heterotrophic bacteria are very varied in their function. They will consume sludge, they will consume cellulose, fats, lipids, oils, starches, nitrate, phosphate, ammonia, nitrite, all sorts of stuff. Heterotrophic bacteria in general are heterotrophs just like we are. We need to consume organic carbon in the form of our food as fuel. Bacteria need the same, but they need organic carbon in a very different way, in the form of fatty acids or ethanol or acetate, or essentially organic carbon from an organic source, whereas the nitrifiers use inorganic carbon to consume their food. Tim, how much part per million of ammonia is too much? I would say that if you're above 10, stop and let it come down naturally. I would aim for 4 to 6 at a maximum. Okay. So they take organic carbon and they play a little game with their food. They reduce that to other less complex base forms. So let's take denitrification, for example. Now, there are two sorts of denitrification. There's assimilatory denitrification and dissimilatory denitrification. Assimilatory denitrification happens in an aerobic oxygen-rich environment, which is where biopellets live, biopellets in marine systems, or where you're dosing an organic carbon source like NO3P or 4X, vodka being a sugar, anything like that, into your aquarium, which also fuels dissimilatory denitrification as well. So what happens with assimilatory denitrification is the bacteria that are floating around in the water column are accessing the organic carbon that you're either putting in, or that they're living on that organic carbon in that biopellet. And they're taking nitrate and they're taking phosphate and they're incorporating it into their cell structure, and they're dividing and multiplying. That's then getting exfoliated, or it's floating around in the water, and your skimmer rips that out and therefore harvests and removes it from the system. Okay? Dissimilatory denitrification is only taking nitrate and turning it into nitrogen gas. It is still using organic carbon. It is using the nitrate as an oxygen source, breaking the nitrogen off, and through a couple of different processes, transferring it from nitric acid, nitrous oxide, nitrite in some cases, and then to various other different processes into nitrogen gas, and that is gassing off with your water circulation and water movement on the surface of your aquarium and so forth. Different sorts of heterotrophic bacteria will live in different areas of the tank. It's not only denitrifying bacteria that live in the anaerobic zone within your aquarium. Different sorts of heterotrophic bacteria, whether they're good or bad, will live there, and aerobically as well. Disease-causing bacteria can be heterotrophic bacteria, and in most often cases are. Just because you're dosing a probiotic heterotrophic bacteria doesn't mean that you're dosing, pathogenic or unwanted bacteria into your system. So their metabolism, again, is fairly simple, but it is very, very varied. Organic carbon, their food source, and then they reduce it and oxidize it into a form that they can get the things they need out of it and break it down into, more simple forms that they can use, that other bacteria can use or do use. the manipulation of water quality and nutrients for different outcomes. Okay, so manipulating water quality for heterotrophic bacteria is for different outcomes. Specifically, I'm talking about manipulation of the Redfield ratio and the reduction of both nitrate and phosphate within a aquatic system. So we're looking at adding organic carbon where organic carbon would usually be limited to a system to result in the enhanced uptake of ammonia and nitrate and phosphate within the system. So by adding organic carbon, which are usually organic carbon limited, you allow that bacteria to proliferate. Now, you need not only organic carbon, but you need nitrate and phosphate for that reaction to occur. Now, in more often than not, you will become nitrate limited because you need, look, I'm going to throw numbers out here, 50 parts per million of organic carbon to 10 parts per million nitrate to 1 part per million phosphate. You generally have a shitload more nitrate than you do phosphate, but phosphate is a little bit harder to remove, yeah? And it binds to calcium carbonate within the system, which is your substrate and your live rock and all sorts of stuff. So you can get phosphate coming from seemingly nowhere where you didn't have a very low level before, but it's actually coming out of the substrate and out of the rock from high concentration within that rock to low concentration in the water. And that's, when you're dosing organic carbon, when you're getting that movement from high concentration to low concentration from your substrate at the substrate water interface, and substrate I'm talking about now is gravel. That's where you'll get cyanobacteria and hair algae growth and things like that, as well as on your rock and stuff, because it's grabbing the phosphate as it's coming out of the rock and out of the substrate. Yeah. So where we have that happening, you can reduce all the nitrate in your system, and because nitrate doesn't bind to anything, it just floats around in the water. So once you've got rid of it, you've got rid of it, right? And you're just having the nitrate coming in from your feed. You get rid of all the nitrate, and all this phosphate is bound up in your substrate and your rock, and you're all of a sudden nitrate limited. What do? Well, you could add something like potassium nitrate, but if you go around asking for that, people are going to think you're going to build bombs. I use Seachem Flourish Nitrogen, which has urea and nitrate in it, very effectively, to the point where I had exactly that problem. I had a loaded phosphate problem where phosphate had not been controlled in the tank for, I'm going to say 5 years, maybe more. And I got rid of all the nitrate, and all of a sudden I had these levels of like 2 to 4 parts per million phosphate. It was ridiculous. I was adding Seachem Flourish Nitrogen in the morning, dropping it to 0 phosphate, and by mid-afternoon it was back up to 2 to 3 parts per million. That removal of the phosphate by the bacteria, it was getting stripped out, and then it was at a low concentration in water and high concentration in rock and in the substrate. So that gradient meant that the phosphate moved out back into the water, and so you had this removal in the water, phosphate coming out of the rock, removal in the water, phosphate coming out of the rock, and that's how we got rid of it. We went through a shitload of product to get rid of it, and in hindsight it would have been easy just to rip all the rock and the substrate out and start again, but proved the point. Seachem Flourish Nitrogen. Brightwell Aquatics actually have a nitrogen supplement, as well as I believe Continuum also have it. Some German reefers actually use ammonium chloride, like I was talking about with fishless cycling, because ammonia gets turned into nitrite, which gets turned into nitrate. Just increase the load on your biofilter. As long as your biofilter is big enough to handle it, you won't have any ill effects on it. You do have to be a little bit careful. But, yeah. So that's how you can manipulate water quality. Now, you can also become phosphate limited, and that means you need to add a little bit more phosphate to the system. What do you do? If you've got that problem, congratulations. It's a lot easier to fix the nitrate limitation if you've got a phosphate-loaded system. The other thing is increasing oxygenation within your bottom sediments so that the heterotrophic bacteria can break down the waste sediment products within the system itself. So making sure that you've got good circulation, good oxygenation. Obviously, I don't recommend that if you're running a deep sand bed. Don't go oxygenating your substrate or reducing it to increase the flow through your substrate because you're going to mess that up. So yeah, but thanks, Tim. Do escarot shells leach phosphates? I don't know exactly what that is. Can you explain what an es, escargot? God, yeah, what that shell is. I'm assuming it's a snail shell. I would imagine that that it is made of calcium carbonate, so I wouldn't imagine that it would leach phosphate. Would it bind phosphate if phosphate was at a high enough level? It's made of calcium carbonate, so unequivocally, yes, it would. If you had elevated phosphate levels within the system. It's not going to leach phosphate if phosphate was never there, because phosphate isn't incorporated into it when it's made. But if phosphate is present within the system and then those shells are in there, yes. French snails that you eat, why are you putting those in your aquarium? I don't think the problem is phosphate. I think the problem is that you're putting edible snail shells into your aquarium. That's the first problem. But yeah, same rule applies. Chicken shells, they're not going to leach. I mean, arguably, probably they have some sort of phosphate in them, but yeah. Oh, you have shell dwellers. Okay, that makes more sense. Look, I can't say that they will or they won't straight out of the packet or whatever it is that you have when they come dry. Yeah, I can't say that for sure. You'd have to break it up and do a Triton test on that. But yeah, if there's phosphate in the system, they will absorb it if it's at high enough levels. If dosing ammonium chloride and monitoring the parameters, what levels of ammonia and nitrate would you become concerned with or slow down in a marine tank in the process of trying to raise the nitrate? We've talked about this, Zoe. Thank you very much for the loaded question. If I was dosing ammonia in an effort to raise nitrate, I would be concerned if the biofilter was not keeping up at any stage. So starting low and working your way up, because you've obviously got to start low. I mean, even though your biofilter might be huge and it might only be operating at 30% capacity, you've still got to nurse the bacteria till they grow that level. If you've got any detectable levels of ammonium and nitrate whilst you're artificially dosing ammonium chloride to result in an elevated nitrate level to lower phosphate, like we've talked about, then that would concern me. I would be concerned with levels of 0.25 parts per million of ammonia or 0.125 to 0.2 parts per million nitrite. In salt water, nitrite really isn't an issue because of the amount of chloride in the water. But yeah, if you've got levels, slow it down. Like we've talked about, if you can find a nitrate supplement that is just dosing nitrate, then you're going to be 2 steps ahead of the game, literally, already. In a moderately alkaline aquarium, they should be reasonably inert. I would argue the same, but that still doesn't mean that they won't absorb phosphate if phosphate is at an elevated enough level. Yeah, so, yeah. Sorry, I need refreshment. Hope everyone is enjoying beverages. Cheers. Oh, come on, Tim. You'll go to saltwater one day. It's all a learning curve. No one's an expert when they start. Trust me. You just got to start. In some ways, saltwater is actually easier than freshwater after you get over all the equipment and the dosing you need to do, because there's only one set of parameters that you really need to worry about, and then you sort of move outside of those depending on what you're keeping. So Guinness, well, it's a stout. It's not Guinness. It's a stout, but it's not Guinness. No. Manipulation of water quality we've been through. Use in aquatic systems we've pretty much been through. That is for reduction of waste products. Now, the other thing I haven't gone over is reduction of disease. Now, there's two main modes of action that probiotic bacteria have in reducing disease prevalence within a system. Firstly is competitive exclusion. So essentially you're dosing it to such a level that there is so much beneficial bacteria in the system that, one, the environment is really, really good, so pathogenic bacteria just don't want to be there. And secondly, there's that much bacteria there, the pathogenic bacteria just can't get a foothold in the system because there's no room for the pathogenic bacteria to exist because there's all these really good bacteria in there that are dominating the biofilm. Yeah? There's no, it's like having a really healthy lawn generally won't have weeds in it. Okay? You nurture your lawn, you fertilise it, you mow it regularly, you get out there and sing songs to it, it won't have weeds in it. Okay? So that's one way. Pretty, there's nuances to it, but that's pretty much it in a nutshell. Secondly is the production of substances that are antagonistic to disease-causing organisms. So I'm talking about antimicrobial, antibacterial, antifungal, and antiviral substances. So that I talked about Bacillus subtilis before. They produce a product called subtilin, which is an antibiotic, an antimicrobial, that is very, very, very effective in suppressing Vibrio. Vibrio has been heavily linked to a lot of bacterial infections as the primary bacterial infection. That's why aquaculturists test for it with plating very, very regularly, because it's a precursor to a lot of other diseases, as well as it causes havoc by itself. Strangely enough, Vibrio has been theorised as being one of the main causes as RTM and STM in SBS corals as well. One of the causes, not the only cause, one of the causes. Bacterial RTM and STM. Other ones are there's a lot of different antimicrobial, antifungal, antiviral, antibacterial substances that are produced. A laundry list of them. Bacillus subtilis, to give you an idea of how effective it is. In World War I or II, I can't remember which one it was, where they were training in Egypt. They had these Bedouin guys that were going around, and the soldiers were getting dysentery, and they didn't know what was going on. And these camel riders and Bedouins were getting around with them, and they weren't getting any of this problem. They just don't suffer from this disease. It was going rife through the camps, and one day the soldiers actually witnessed the camel defecating on the ground, and these camel riders went up and started grabbing handfuls of camel droppings and started chowing down on them. And they're just like, they got the translator, just like, oh, it stops us from getting sick. That's really all they could get out of them. Okay, so they take some of this camel dung and they send it to, a scientist, to a lab, and they isolate this bacteria, which would later be known as Bacillus subtilis. Now, the Bacillus subtilis strain not only has the ability to regenerate cells, it has a probiotic, ability to regenerate cells, but through the production of subtilin and other antimicrobial substances and stuff like that, it was able to fight off dysentery. So they started mass producing this in a powdered pill form and feeding it to their soldiers, all of a sudden, no more liquid poo-poos, no more tap ass. So there you go. And you know. So yeah, what the production of antimicrobial substances does is it essentially, you know, fights off gram-negative and pathogenic bacteria, producing natural antibiotics that can help to reduce disease. And as a lot of you will know, most parasitic diseases and viral diseases actually start as a bacterial disease, as they, you know, that's the first thing that kind of sets in an open wound or anything like that. Then your the immune system of your fish gets compromised, and that's where you start to encounter a lot of problems. So nitrate and phosphate reduction in marine and freshwater systems. We've been over marine systems with assimilatory denitrification. Arguably, you could also do it in freshwater systems as well if you had a way of removing that. Freshwater protein skimmers do exist, probably not at an aquarium level, but I use them all the time in aquaculture. Very effective. Very, very effective. So dissimilatory denitrification is able to be achieved in freshwater systems through the use of different media that harbour oxygen-free zones within them that allow denitrifying bacteria to exist. With any metabolic process with bacteria, even nitrifying bacteria, you're going to get phosphate removal. Whether it's substantial enough to control high levels of phosphate within the system has remained to be seen. I haven't seen any research on that in particular. So, yeah, it's pretty much the same between freshwater and saltwater systems. Freshwater systems is a little bit harder to control because you don't have the removal mechanisms that you do in saltwater systems. So, in regards to dissimilatory denitrification, we've got a process where you have ammonia production and ammonia consumption. And when you have ammonia consumption, you've got alkalinity consumption at the same time, right? That gets turned into nitrite. So it's oxidized and it has alkalinity added to it. You then have nitrite accumulation and nitrite reduction. And when you have that nitrite reduction, you have further alkalinity consumption and oxidation of that nitrite. It turns it into nitrate. And then you have nitrate, and it climbs and it climbs and it climbs and it climbs and it climbs. It's been oxidized twice from ammonia to nitrite and nitrite to nitrate. And each of those times, that bacteria has used alkalinity to do that. When you get dissimilatory denitrification, so you have nitrate in an oxygen-free zone with heterotrophic bacteria that are taking that nitrate as an oxygen source, breaking the oxygen off it and getting nitrogen gas out, the alkalinity that's been used in the ammonia oxidation and the nitrite oxidation is sequestered back to the system because the nitrate is broken apart. It's not broken apart by an autotrophic bacteria. It can't be oxidized any further. Nitrate is as oxidized as the nitrogen ion is going to get. What happens is the bacteria break off the oxygen from the nitrate ion to respire. They take the organic carbon out of the system to feed on whatever it is, the other stuff that they're feeding on. Nitrogen gas is a byproduct, and the alkalinity has nowhere to go because heterotrophic bacteria, we know, don't use alkalinity. They use organic carbon, fatty acids, ethanol, vinegar, sugar, all of these things that are broken down from organic forms of carbon. Okay. So then you have alkalinity come back to the system. So denitrification will actually sequester alkalinity and add alkalinity back to your system, which means you won't have to add alkalinity supplements as much, or you will see a rise in alkalinity. So, for instance, in a marine system that has very high nitrate levels, that has no denitrification occurring that you can see or that you think is there, and all of a sudden you've got a huge protein skimmer on your system and you start adding heaps of organic carbon to the system and your nitrate level drops from 80 parts per million to 0. If you were to continue with your current dosing regime of alkalinity, I will almost guarantee you that you will see a rise in alkalinity, whereas it would have been stable previously. And that is simply because the alkalinity that's being fed back to the system is going to be over and above what your alkalinity is that you're maintaining as a constant level. It happens the same in fresh water. It's happened the same in the data that we've seen from the testing that we've done on the biofilters that we've cultured recently. Okay, so we've seen ammonia go up, down, nitrite go up, down, nitrate go up, and then peter down, and sort of stay low. And as ammonia and nitrite are being consumed, that nitrate's staying low, but the alkalinity continues to go up. Very, very, very interesting. A very interesting phenomenon to witness. Not a phenomenon, a very interesting biological metabolic process to witness and see. So there's actually a ratio. I think it's for every gram of nitrite that's denitrified, 7.18 grams of alkalinity is sequestered back to the system, something like that. It's quite substantial. Which is why denitrification systems in aquaculture are very useful, because not only does it reduce the amount of water that you need to exchange, it also allows you to anaerobically digest sludge, as well as sequester alkalinity back to the system. And when you're putting a ton of food into a system a day, alkalinity becomes a very, very, very large part of your dosing regime. As we've talked about. Food in equals ammonia in equals alkalinity out. So if you can get some alkalinity back by denitrification, why not do it? And your fish will love you for it too. Lower nitrate levels is shown to be very beneficial for fish. Okay, so aging systems and the effect of the microbial herd diversity. Aging systems, or old system syndrome, or old tank syndrome, you'll often hear people talk about. In a closed system with no input of outside bacteria, obviously you're going to have bacteria floating around in the air and all sorts of stuff like that, that are going to settle into the aquarium and add to the microbial herd. But essentially speaking, unless you're putting water from the environment straight into your aquarium, you're not going to have a constant input of diversity of microbes in your system. And you could argue that could actually be dangerous, because you don't know what microbes or organisms you're actually putting into your system. Okay, so I was talking about the evolution of biofilter bacteria, and how I said that even if you add very, very efficient strains in at the start in a concentrated form, an isolated form, would 2 to 3 months down the track, would they be the same bacteria in the system? Probably not. You'd have other bacteria in the system. You'd have, whether or not you'd have that bacteria still residing within the system would be questionable. The system becomes a constantly evolving organism in itself. What does happen, especially in the case of heterotrophic bacteria, because they produce at a rate of knots higher than nitrifying bacteria do. Heterotrophic bacteria will not only continue to metabolize and divide and multiply every 15 to 30 minutes, but as they do, and they make a copy of 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 a copy, they become mutated and they become less efficient at what they're originally meant to do. So what happens is you get single species of bacteria or a dominant species of bacteria within the system that isn't really performing all of the metabolic functions that you need in your tank to keep it healthy, right? That's where adding a probiotic bacteria mix can help to reinvigorate your microbial herd. Okay, there's other factors in there, like obviously you go through and do a gravel siphon, you do a filter clean and stuff like that. Not in tap water, in tank water or aged water with no chlorine in it. And then you would add probiotic bacteria to help to reinvigorate the microbial herd and the microbial diversity within your aquatic system. Okay, that will add back different bacteria that are doing different things. It'll improve the diversity within your system, and it'll, get your system back on track. Variety is the spice of life here. So, you want diversity within your microbial herd. You don't want single species or dominant species within your system because they're not performing all of the functions effectively of what they could. Yeah. Paul. Some people refer to Oltek syndrome as the buildup of anoxic bacteria. Okay, so there's a couple of different terms that we need to get out of the way here. Oxic and anoxic are the environmental conditions. So oxic means the presence of oxygen, anoxic means the lack of oxygen. They are an environmental term. Status, if you will. Now, aerobic and anaerobic is the process of the bacteria that live within those different circumstances of oxic and anoxic. Okay, so anaerobic bacteria that live in the absence of oxygen live in an anoxic zone. Aerobic bacteria that live in oxic conditions in the presence of oxygen live in an oxic zone. Okay, so by anoxic, I'm assuming you mean anaerobic bacteria, or in oxygen-free zones that don't have levels of bacteria within the system to break down waste. So what I would say is the build-up of anoxic bacteria. I would more say the build-up of an environment that is free of oxygen, that doesn't promote aerobic bacteria that can break down the waste that is within that. So the waste continues to build up and continues to build up and continues to build up. And when you have excess waste and areas that have no oxygen in them, that, you're really only going to have denitrifying bacteria and pathogenic bacteria hanging around in, what's going to happen? You're going to have excess waste within the system. You're going to have nutrient cycling. You're going to have all sorts of problems with ammonia nitrite breakdown if those waste products get disturbed. You're going to have disease-causing organisms at a much higher prevalence. So not so much the build-up of anoxic bacteria. I would say the build-up of an anoxic zone within the system from a lack of maintenance, that would arguably have anaerobic bacteria in it that may not be beneficial to your system. So, yes, I would agree with that, but there's a deeper dive into it, that it's not quite as simple as that, but it hits the nail pretty much on the head if you look further into it. So, yes, Paul, I would agree. Could seeing alkalinity rising in rivers potentially happen due to the use you've just been talking about once nitrates steady out? Okay. So, if you had, well, look, see, sequestering alkalinity is going to happen only in an aerobic dissimilatory denitrification system. Okay, so bacteria that are living in an aerobic or an anoxic, that are anaerobic, that are living in an anoxic zone within the system, that are breaking down the nitrate and converting it to nitrogen gas and sequestering the alkalinity back to the system. Once the nitrates steady out, it's possible, but I would probably say that you would have to have a metric shit ton of nitrate going into the system to notice any significant rise in alkalinity because of the constant flushing of the system, as well as the physical dilution rate and volume of water that you're seeing within the system. If that was stagnant water and it was a pond and you got enough dissimilatory denitrification to fuel that amount of alkalinity sequestering within the system, then perhaps. My thought would be that if you had excess runoff from agricultural land with excess nitrates within the system, that then went into the system, you would actually see algal blooms before you would see any level of denitrification from bacteria and sequestering of alkalinity. And for it to sequester alkalinity, my argument would, look, if it's nitrate ion that's going in, it may have been nitrified within the soil system. So yes, it would probably add alkalinity. Hence why agriculture farms add lime to their system to enhance nitrification to get nitrate out of organic matter that they put in. Continuous sludge accumulation, yeah, Bruce, yeah. So hit the nail on the head. I understood anoxic is when oxygen is not present, even in nitrate or phosphate. Okay, so anoxic is when oxygen is not present. Correct. It is when oxygen is not present. It is the environment. So there is no such thing as anoxic bacteria because anoxic is describing the environment, not the process of anaerobic metabolism that the bacteria live in an anoxic zone. Understood anaerobic zones are when oxygen is not free but only present in nitrate and phosphate. I thought anoxic is with oxygen, is not present in any form. Thought anoxic is with oxygen, is not present in any form. Okay, so the easiest way to think about it is anoxic is an environment where there is no oxygen. Anaerobic bacteria, so not aerobic, no air, live in an anoxic zone that has no oxygen or very little oxygen. Whether or not there's nitrate and phosphate within that environment doesn't matter. It's the zone that you're describing when you talk about anoxic. Anaerobic zones, yeah, okay, you could use that description as describing that zone as well, but anaerobic is more the description of the bacteria that are performing a metabolic function anaerobically as their metabolic function, not as a description of the zone that they're living in, which is anoxic. So oxic, oxygen, anoxic, no oxygen. So, yeah. Okay. So who here runs biopellets or doses organic carbon actively within their system? And do you find that you clarify that as to dissolved or ionized oxygen, I think is where Paul's question was. Clarify that as to dissolved or ionized oxygen. Okay, so an oxic environment or an aerobic environment is in the presence of oxygen. If you don't have oxygen, it's an anoxic zone, and anaerobic bacteria live within the anoxic zone. What triggers hydrogen sulfide? Hydrogen sulfide is the product of a bacteria breaking down waste without the presence of oxygen, where there is a buildup of organic matter. Okay? So if we're talking about a healthy anaerobic zone, we're talking about a healthy environment where you've got water flowing through, you've got all of the oxygen being consumed by, say, you've got this big porous block of filtration media. Paul, you know the stuff that you really love, that marine pure? Say that you've got that big block, right? And in the first 5, 10, 15, 20 mil, you've got aerobic bacteria that are processing in the presence of oxygen ammonia, nitrite, different waste materials. And then all the oxygen's been consumed, and on the inner parts of that brick, you've got no oxygen, but you've still got water flow, but you've got no waste either because of that very porous environment where it's very clean. You've got no waste, but the transport of nitrate and phosphate and other materials into that environment where there's no oxygen, but the bacteria living in there are really only exposed to very low levels of mold or no levels of mold and nitrate and arguably phosphate. But let's just talk about nitrate for the moment with dissimilatory denitrification. So breaking that nitrate into nitrogen gas and alkaline and whatever. So you are harboring a very specific group of bacteria that are just denitrifying nitrate. Okay? If you were to take that marine pure block and you were to grab that block, stick it in your sump, and then pour a heap of substrate over it, dirt, gravel, whatever it might be. And as time goes on, you've got mold that's settling within that substrate. It goes down. If you don't have oxygen going right down, you've got like a 6-inch, 8-inch deep, media bed there. And you've got waste building up, and you've got bacteria in there that aren't denitrifying nitrate, they're just eating stuff. A byproduct of some of those bacteria can be hydrogen sulfide. They're not beneficial. They are producing a reduction process, but they're producing a byproduct that you no longer want. And it's because they're in the presence of a lot of mold and a lot of just stuff that breaks down and just turns into nasty gear, and it's just you're not, by eliminating the solids within the system, you reduce the ability for hydrogen sulfide to be produced because you don't have that organic component in there that's going to proliferate the bacteria that are going to produce that hydrogen sulfide. That's where these porous sorts of medias shine, because they can produce a oxygen-free environment or an anoxic environment for anaerobic bacteria to reside, but they're not going to proliferate harmful bacteria that produce hydrogen sulfide through the breakdown of solids. I hope that answers your question. Please explain mulm. It was common knowledge but lost. Mulm is pretty much just waste. There's good mulm and bad mulm. Good mulm is mulm that's been broken down, thin layers. You've got all sorts of different microorganisms that are hanging around in there. In a planted tank, arguably, you would have a lot of mulm in there. The plant roots are going to get in there. They're going to oxygenate it. They're going to break it down into humus. And the mulm is going to be healthy. Bad mulm is mulm that's essentially just chunks and chunks and chunks of build up of a hell of a lot of fish shit. And that's bad mulm. So mulm is usually it can be mineralized or oxidized, or it can be bad stuff that's broken down anaerobically. It's still, it's still mineralized, but that breaking down of that mulm by anaerobic bacteria is what's creating the hydrogen sulfide. So you want to do it in an aerobic, oxygen-rich environment, so those heterotrophic bacteria that are grabbing that mulm and grabbing those waste products and breaking them down in the presence of oxygen, they're not producing any of these awful byproducts, the hydrogen sulfide. They're just producing CO2 and water and breaking it down into more organic carbon for other heterotrophic bacteria to use, and they're grabbing their starch or their fats or their lipids or their cellulose or whatever it is that makes them happy, and just getting rid of less simple compounds that they've broken it down into, and they're using enzymes that they produce in order to help them do that. Mulm is very important when it's aerobically produced, because you're going to have all sorts of critters that are going to live in there. You've got bacteria that are going to live in, biofilms that are going to attach in the mulm and clump it all together and stuff like that. If you had a bare bottom tank and you've dosed probiotic bacteria in it, you probably notice all the waste and stuff actually clumps together in a big clump if you've got probiotic bacteria that's actually doing a good job. Or they'll break it down completely. Yeah, so mold is important. Mold is important as long as it's the right mold. Look, you're going to have mold in all of your tanks. If you have a healthy substrate system, say in my tank, which is terrible and neglected, and I've got to get rid of it because it doesn't match the furniture in the new house. So it's got to go. If I get my hand in there and I dip it in, it's probably only about, an inch thick or something like that. If I get my hand in there and move it up, I get a little bit of waste come out. But it's very fine, it's broken down, it's not black, it doesn't smell. My tank doesn't have that awful old stale water smell. Because I have quite large particles, I have good turnover, and I dose probiotic bacteria into my substrate. I very rarely clean my substrate. I'm quite lazy, but it doesn't need to be cleaned because it's low enough to create oxygen diffusion through the system. And I've got, healthy bacterial, biological population within there that help to break down that waste. So every tank is going to have mold. Every tank is going to have mold. It's just the use of that mold that's fallen out of fashion because there's better ways of doing it, like adding probiotic bacteria or using a better filter or, anything like that. Tim, yeah, no worries, Tim. Hope to see you a little bit more active on the group. By all means, if you have any questions, we'd be quite happy to field them, either privately or publicly on the forum. So thank you very much. I thought mold was sediment totally broken down. It can be, or it can be sediment that isn't broken down. Good mold is sediment that's completely broken down. Bad mold may be partially or completely broken down, but by bacteria that you don't want breaking it down. If you have mold within your system that comes out black and it smells and stuff like that, wrong type of mold. Good mold is mold that, we've said mold way too many times. Good mold is mold that is broken down, which could arguably be then classified as humus that's organically mineralized. If you ever look into, if anyone ever has had a worm farm, worms have the fantastic ability not only to completely change the microbial diversity from negative to positive, like for instance, worms will absolutely eviscerate E. coli populations. In their gut, they can physically change that. They can change the microbial diversity from negative to positive. But the bacteria and the stuff that they feed on enables them to take organic matter and turn it into very, very highly mineralized, very highly broken down and stable forms of humus. That's why when you put worm castings into soil, or dare I say it, into an aquarium with some people that do planted tanks, they've used worm castings in the past, their plants absolutely thrive because it's completely broken down organic matter. There's no longer mold, it's humus. And humus is what you aim for in your pastures and in your farming because it adds organic carbon back to the system. It adds all these different oxins and all these different byproducts that plants really love. Mm-hmm. Geez, there's too much information, Tim. Go and have fun. Detritus, mold, humus, they can probably be all grouped into the same thing. Detritus can also be grouped into stuff that isn't broken down yet. So detritus could be just leftover food and shit and stuff that's not broken down. Detritus could also be solids and dust and particles and stuff that is comprised of mold and different things like that. Yeah. Bad mold each time you eat curry. Well, maybe it's cleaning out the bad mold, Bruce. Maybe it's cleaning out the bad mold. Maybe you need to take a probiotic injection of whiskey. That might help to rebalance your system. Detritus, yes, it does break down into mold. So detritus is usually characterized as the stuff that isn't broken down yet. Yeah. God almighty, let's not, we want to keep this group a bit classy, Paul. So, keep your shirt on. No linking your Pornhub account. And let's just, cancel. No, what am I doing? Guys, I've got notifications and, here we go. Okay, so no one is actually wondering how to get in. That's fine. Okay, any other questions? It can, and you know what, Bruce? As much as we're having a bit of a laugh about this, that's very, very, a very apt description. It smells anoxic and smells yuck with that, really bad smell. And you know why? Because curry, chili, garlic, the amount of spices and things like that in there, they're creating an effect of scouring your insides. So if you have an imbalance of bad bacteria and good bacteria in your gut flora, eating things like that have actually been shown to be beneficial of ridding you of internal parasites and bad bacteria. So just like you do when you have a foam fractionator and you've got a heap of bacteria in your system, you ever notice how when you dose organic carbon into a marine system and your skimmate all of a sudden starts stinking like a piggery? If all the stuff is left there to break down, that's dead bacteria. So, after you have, something that scours you out like that, or scours an animal like that, it's physically the scouring of bad bacteria that's coming out and breaking down. You're smelling bacteria farts and bacteria decomposing. That's exactly what you're smelling. So, yes. Hydrogen sulfide only smells within certain levels. The human nose can detect hydrogen sulfide levels as low as, I think, 0.01 ppm. So it only smells within certain levels, yes. Once it gets to high levels, I believe that it then stops smelling, and that's when it's very, very harmful. Hydrogen sulfide's bad business, man. Bad business. There was actually a massive fish kill at a salmon smolt production facility in Europe that was specifically related to hydrogen sulfide and the release of hydrogen sulfide from a large gas bubble that had been building up. This is why I use moving bed filters as opposed to static filters. Now, some of these, it's not older technology, but it's technology that some companies use, and if it's maintained, it works very well. But essentially, it's moving bed media that they pack into a bed and they flow water through. It removes a lot of fine particles, and it does do very, very effective biological filtration. But if it's not maintained, that's where you get a build-up of all this waste product and where you can get this hydrogen sulfide-producing bacteria. Now, build up to such a level that then it bursts out of the system and into the water column. 7 tons worth of salmon smolt dead. That's why people shy away from static bed filters now. So it's very, very dangerous. Very dangerous stuff for fish and for people. Andy, what, if any, bacterial products do you recommend for reef aquariums? Okay, so, all right, this isn't a product recommendation stream. With the products that we've developed, we have used them, and they are currently being used in reef aquarium systems. And the feedback that I'm getting, they're getting some good results. The products that we sell and we develop, we sell in a minimum of litre bottles. We're not involved in the aquarium industry at all. Okay. The products that I have used in the past, I find the product Microbacter 7 from Brightwell Aquatics to be very good for aquarium use. I have used Zeobac and Zeozyme, and I've found very good results with that as well. Some of the new probiotics I haven't used over and above that. I haven't used any of the Continuum products, but the fact that Continuum owns Brightwell and that a lot of their products actually cross over and pretty much are the same on the face value of the label, I would imagine that some of the Continuum products may possibly be just as good as Microbacter 7. I can't confirm that. I have no experience with that, but it might be something that you want to look into. The thing that you need to remember is bacteria, even heterotrophic bacteria, spore-forming bacteria, have a shelf life. If they're a powder, they're going to be more stable. If they're a liquid, if they're a nitrifying bacteria and they're a liquid and they're kept at room temperature, after 6 months you're buying very, very expensive water. And if they're imported, you got to remember that they're produced, then they're bottled, then they're shipped over by sea. Shipping by sea can take 6 weeks. Depends on how long that they've actually sat on the shelf there as well. All I'm saying is look at your bacterial product and the age that the bacterial product is. Try and get something that has a high turnover and something that preferably is refrigerated, whether it's a probiotic or a nitrifying bacterial blend, preferably something that is refrigerated. Refrigeration will improve your bacterial product out of sight. Yeah, you're right, Bruce. It does get desensitized quickly because she knows physically can't handle it, from what I understand. Okay, so you use aquarium detox to rid hydrogen sulfide. I would argue that you're using aquarium detox to rid the waste from the system that the bacteria that produces hydrogen sulfide would produce. So you're not physically removing hydrogen sulfide, you're actually creating an environment that doesn't allow bacteria to proliferate that would produce hydrogen sulfide. Polyfilter removes sulfide. Okay, cool. You have microscope tested many bacterial plants and brands, and many don't have bacteria in them. I can't say that I'm an expert on microscope slides, but I believe you would probably need to stain them to be able to do that and need quite a high magnification, unless you're getting a lab to do it. I can say with our bacterial products, the reason that we went to isolating and developing them within Australia is because, one, we have a very high turnover of our bacteria now. In our system, our bacteria maybe stays in our fridge before it's sold for about a month, and we can get our bacterial products from the time of placing order to the time of receiving our order is usually about a week and a half. So even though we're out of stock, our bacteria is at most about a month and a half old by the time we sell it. It's very fresh, very high cell concentrations, and we base all of our dosing regimes off that. We've developed a dosing regime over the years that allows us to sort of dial in what sort of dose you need to achieve a net positive result for what you're trying to do. Channel 9 did in the lab, Jesus. I don't expect you to mention names here, but if we're ever chatting, Paul, it'd be nice to have a chat about that. It'd be interesting. Not that I'm interested in knowing, what brands have bacteria in them or what don't. It'd just be interesting to see which ones are imported as opposed to which ones are local, and see what the differences are really. Standing at all emotion magnification levels for a lot of bacteria. Yeah, okay, Bruce is the expert. I don't claim to be an expert in microscopes. I actually hate using microscopes. I can never get them to focus. My eyelashes get in the way and all sorts of stuff. I hate using them. I'd rather use a microscope that has a camera on it because then my head has to go nowhere near it. Really? Okay. Polyfilter. Look, polyfilter. There's been a lot of work done with polyfilter. A lot of people say, "Oh, polyfilter, it's expensive." Blah blah blah. Yeah. Look, it's been it's been used time and time again, and positive results have been seen with it by using polyfilter. I haven't used it really at all personally, but look, having said that, guys, the people who use it wouldn't be buying it and continually using it if it didn't work and they didn't believe in it and they weren't getting results from it. They wouldn't be paying the money for it or bothering to stock it and sell it in their stores if they didn't see benefits in using it. Even if it's not even the primary filter mat in your system. And arguably, for something like polyfilter or one of your loaded filter mats like a zeolite filter or a carbon filter or anything like that, even activated carbon or zeolite in the filter bag, arguably, you probably should be pre-filtering it. It will remove the solids from the system that are going to clog the filter, that are then going to break down in the polyfilter and potentially load it faster and get you less effectiveness out of it. So if you've got an aquarium store that's selling you polyfilter, do yourself a service and go and get some filter wool, or better yet, some cut to size, just normal filter mat, filter foam mat. Like your 10 or 25 mil filter foam mat, just your white stuff that you can throw away. It costs a hell of a lot less. And you're going to protect and enhance the lifespan of your polyfilter. So, it's the same as pre-filtering your biofilter, or pre-filtering it before you run it through, like pre-filtering your canister filter before you run it through your biological and chemical filtration, like your carbon. It's a chemical filtration media. Oh, really? Was it? Just after blood transfusions. There you go. Well, if they're pumping human blood through it, it's got to be good for fish tanks. Don't want to be pumping it through something like that, pumping blood through something like that, and then putting it back into your body and hoping that it all goes well. So I've got a message from David by the look of it. David, yes. After the stream, I will send you. I'll look. Facebook message me your email address. I will then email you from my work email that's got my phone number on it, and please call me at a time convenient for you between the hours of 9 and 5, and I'll be quite happy to shoot the breeze with you, mate. I actually do go on parental leave at the end of this week. This week might be a little bit busy for me, actually. I'm trying to frantically scrambling to get things organised. I go on parental leave for 10 weeks to look after the young ones. So, within that time, I could even talk during the day from next week onwards. We could sort out a time to call, or you could send me your number even, and I could give you a call at a time convenient for me. Obviously, with a 4-month-old, there is no convenient time, so to speak. It just happens when it happens. So, yeah, just let me know. Let's not try and cover too many subjects at once, though, eh? Pick one, and then we'll go from there. Even email, if you have questions to ask, I can answer your questions by email as well. Dacron by the meter at Spotlight is a very good place to start, Bruce. Definitely. Definitely a good place to start. Does anyone have any other questions? Any publication lists you've got around the research? As in probiotics in particular? Bruce? Because I've got quite a few publications on probiotics that I could send you, white papers and so forth, that I could send you. You have my email, actually, Bruce. So, yeah, any and all information that you ever are wondering about, you know that I'm available by phone and email at any time to you, mate. Quite happy to just to supply any information that you would be interested in, need, want, need help with, potentially. Yeah. So, does anyone have any other questions at all? Probiotics and also general nitrification. Yeah, I can. Yeah, I can do that. Daniel, you said yes, please. Yes, please to what, Daniel? Can you please explain? I may have missed a comment earlier on up. I'm amazed how much cleaner gravel and filters stay when I use NLS probiotics. Paul, I'm not surprised. I would probably think that the bacteria within the NLS probiotics is probably Bacillus subtilis or something similar. A strain of it, just because it's Bacillus subtilis. There's so many different substrains that do so many different things. You know, yeah. But yeah, the transport of probiotic bacteria to the gravel that then proliferate within the gravel that then create an environment for other probiotic bacteria to be able to proliferate. Yes, definitely. I'm not surprised at all, Paul, and I'm very happy that MLS are bringing out something like that. Probiotics, Daniel. Please send me an email at Gareth, Gareth, at Pure Aquatics, pureaquatics.com.au. With any information that you would like, I'd be quite happy to send it to you. Other than that, you could just go to www.pureaquatics.com.au, find my email address there in my profile under About the Team, and I'd be quite happy to get back to you with any information that you require or need or are interested in. Yeah, look, I mean, I've been spruiking the benefits of probiotic bacteria and manipulating the microbial diversity within aquatic systems for over 10 years now. A lot of people think I'm bullshitting when I talk to them, but then they look at the effects of it and they can't believe their eyes, just like you, Paul. And yeah, you know, it took you using such a great food like NLS that you've been a proponent of since day dot. To be able to do that, yeah, it's of a great benefit. Yeah. So, do we have any other questions at all about anything, probiotics or otherwise? No worries, Bruce. It's an absolute pleasure. What do you think about the influx of dinoflagellates in reef tanks? Usually it is a lack of or a problem with low nutrient. I did actually answer this on the last live stream in regards to water quality. It was in reply to a question that Zoe from Monsoon Aquatics actually had. Is it 5 o'clock somewhere? Definitely is, Brucey. Is Dr. Tim's Eco Balance just to form probiotics? I believe so. Overall dietary needs for Africans, I'm not the person to be asking about that. Yes, I did drink less beer this time. I've been easing up on the beer, actually, and when you're drinking stout, you only need 1 or 2 glasses, and it kind of does you, because it's like I'm eating 3 eggs in every glass. So let's just scroll back up, because I'm missing some comments here, and I don't want to miss anyone. What will my next talk be on, Paul? I'm thinking that we move to life support systems next and filtration, and I believe that we're probably going to have to break it up into several segments. We'll start with mechanical filtration and biological filtration, probably. Then we'll move on to ozone. Then we'll move on to UV sterilization and protein skimmers, probably not in that order. Oxygenation, ozone, that kind of thing. I'll do some work, but yeah, I think we're going to break it up into multiple live streams. 5 o'clock, yeah, Dr. Timms, yes. I used to be cool. Paul, that sounds like a challenge. That definitely sounds like a challenge. Be careful, them's fighting words. All right, so if there's no other questions, and I'm assuming there's no other questions because I've asked the question 3 times now and I've had a few responses, so I'm assuming there's no other questions. I'd just like to take a minute to talk about the current position and status of where we are within our industry at the moment. Look, as a lot of you may know that are involved in the group, the future of the Australian aquatic industry, that Paul has started and that we're continuing to keep the ball rolling on. The AIAA, the PIAA, a lot of industry groups, the Underwater Pet Expo and things like that, we're really gaining some really great momentum within the industry. And like I said before, we're not going to get anywhere unless we work together. It's just not going to happen. I'm going to be completely frank right now. In the past, maybe the last 2 or 3 years, and even before that, but especially the last 2 to 3 years, there has been so much derision and so much antagonistic behaviour between people that it's really just getting old. It's really getting old. There is no point trying to pick out flaws in people's approach to something that are trying to positively make a difference within the industry. You know, I'm going to use the PIAA's formation of the Australian Aquatic Group, or the Aquatic Pet Advisory Group, as it's now called. It's taking a little bit of time to do. We've had COVID-19. We've had Daniel Kimberley, who had to step down because of his work commitments that he had. Daniel Kimberley and Monsoon Aquatics are making leaps and bounds and really accelerating the industry in the realms of aquaculture and coral collection and coral sustainability within the industry, which is absolutely fantastic. And everyone is really trying their best to make sure that we're actually getting a positive outcome here. And yes, it is taking a little bit of time. But up until this point, not a lot's really been done. And we all see this as a turning point to seeing real change. So yes, it is taking a little bit of time. But, you know, there's no point throwing stones and slinging mud at each other, because that's not working together, guys. It's not working together. It's creating derision, it's creating drama, it's creating sour taste in people's mouth. And honestly, it's achieving absolutely nothing except for people getting angry at each other, which further divides us and further exacerbates the problem that we are a divided industry and we're not working together. So just like the last stream yes, time for action, Paul. I totally agree. It's time for action, which is exactly what's happening. And it's time to stop picking fights with each other. Like, we're actually doing something, and once it actually gets done, and once everything is in motion, there's going to be no stopping this industry. But we want to do it. We as an industry want to do it properly. We want to make sure that it's done right so that the same mistakes don't happen again. And fighting with each other and going out of your way to be, and I'm not talking about you, Paul, I'm not referring to you at all, but people going out of their way to be intentionally belligerent is just, it's not achieving what we want, which is a unified industry of people working together. So I'm going to reiterate seriously what I said in my last stream. It's time for us to pull our big boy and big girl pants up and start acting like adults and working together. And just stop trying to use it to accelerate your own personal goals. It is ultimately clear what some people are trying to do. And it's really getting old, and it's not getting anyone anywhere. You know, we're all adults. Let's just work together, for God's sake. We don't get anywhere unless we work together. And this constant, fighting against each other and falling over each other to try and get to the top apple, and it's stupid. Let's just be big boys and girls and try and, act like some semblance of human beings and develop some basic people skills. I'm not even angry, I'm not salty, I'm just disappointed that after all of these, iterations of all of these things happening and all of these cycles of things falling flat on their face because people just can't get along and agree to help each other and help the industry. It's starting to happen all over again, and people are just going, oh, well, something's got to change. We wouldn't be driving cars with round wheels if people, didn't look at it and go, there's got to be a better way of getting around. Like, just come on. It's not rocket science, guys. It's not. You get more with sugar than you do with vinegar, and it's really not that hard to be nice. It's really not that hard. So, I mean, as I said, I'm not angry. I'm not salty. It's got to be said. Enough's enough. Let's work together, for God's sake. Some people are actually working towards that end goal, and some people just seem to want to just cause drama. And it's not even that it's not on. It's just really ridiculous. Let's just work together and act like adults. Otherwise, we're getting nowhere. No, I honestly don't mind if people disagree with me, and I'm quite happy to be proven wrong, but I know I'm not, because I'm getting daily messages from people saying it's happening all over again. I don't want to say anything publicly for fear of being, fear of being reprimanded, like they were in the past, or fear of being shot down or talked to in a way that makes them feel like less of a human. No one should feel like that ever. So if anyone ever wants to, voice their opinion, I'm always here to talk. Let's all get along, honestly. That's all I've got to say on the matter. Does anyone... Good night, Bruce. Have a good night, mate. Does anyone have anything... been up since 3, yeah. The life of a zebrafish operator, yeah. I hope it wasn't too much of a problem that you had to get up at 3 to do that. Does anyone have any other questions? Oh, look, it's not even that, Bruce. It's not even that. It's just the mentality of some people within the industry that are honestly just dragging all the people down that are actually trying to make change and slowing the progress up. So, yeah. And if anyone wants to disagree with me or wants to have a conversation with me or is watching and they think this is directed at them, by all means, give me a call. I'm quite happy to take a call. My phone number is on my email. I'd be quite happy to give it to you if you want to email me. It's not even about haters, Paul, and it's not even about the internet. This is about our industry. So, like, honestly, if you don't, look, I'll put it bluntly, if you don't want to help and you're just going to cause problems, like, don't come to us with problems, don't come to us causing problems, come to us with solutions. And if you don't want to do that, go away. It's getting us nowhere. This fighting and this, you know, he said, she said, just constant battle, getting us nowhere. So yeah, if you want to talk to me, I'm sure you can get my phone number somewhere, or you can email me at gareth@pureaquatics.com.au and I'd be quite happy to talk to you. Send you my number and you can give me a call anytime. I'm not going to put it on the internet, obviously. So yeah, is there anyone that has any other questions at all? It's 12:10. This was actually a really good stream. Hour and 50 minutes on Facebook Live. There was a period of 40 minutes because we started a bit early on YouTube Live, and that kind of went south. So I think I'll just be deleting that portion of the live stream. Yes, but that's you on YouTube, Paul. I mean, you know, like if you weren't being hated, you're not doing the job right. You know, on YouTube, on an aquarium channel, come on. My pleasure, Zoe. I look forward to talking soon. I did talk to Jeff today about the remainder of the equipment. So, yeah, it's getting close. It's on its way. I'll have some more update photos for you soon. And I hope that your nitro problem's going well. Please give me a call tomorrow. Let me know how that's going as well. Yeah, does anyone else have any other questions? I've got pins and needles in my foot. Ugh. Favorite stream yet? Oh well, wait till you I'll wait till the filtration stream. That's gonna be fun. And no, I won't be doing product reviews, I'll be doing technical information. But yes. Marine Pure might just get a mention as a product that achieves a certain process. Go to the PS4, my son. Go with God. Alright, well, it sounds like I've got responses from almost everyone that is responding on the live stream. So, by all means, we had nowhere near the amount of people that said they were coming, but, you know, them's the breaks. That's fine. Yeah. Alrighty then, guys. I am going to bail. Probably go and have a cup of tea. Let's say a good old cup of tea. And I'll talk to yous when I'm looking at yous. Have a great night.